Today I will be discussing about the respiratory physiology, which is a potential important topic for your MB exams. Now, let's begin. First, I will be telling you about the topics that we'll be covering. First, I will start with the basic anatomy, which is very much important to know rest of the topic. Okay. So after that, I will go into the pulmonary circulation. Pulmonary circulation. This thing you have also learned in anatomy, but I will talk a bit about it. And then I will go into the lung volumes and capacities, that is the spirometry. After that, another topic, this I will just touch a bit, okay? Because this thing you will be learning in your anatomy also in the lungs chapter. Here we'll be talking a bit about the mechanism of breathing. Then another important topic which is very much important and mainly asked in your exam, that is O2 and CO2 transport. Okay, this is very much important topic. I will be covering this in detail, and then I will be telling you about the neural regulation of respiration. Means the regulation has two ways of regulation. One is the neural regulation and this chemical regulation. So first I will talking about the neural regulation, and then I will be talking about the chemical regulation. Okay. Then I will covering some important topics, that is the hypoxia, cyanosis, high altitude sickness. High altitude sickness comes in exams very much, so this is very much important. Then I will telling about the decompression sickness. This also comes in exam, which is important. And another interesting topic, that is oxygen depth. This is not important actually, but this is interesting topic, so I will be teaching this also. After this, one question is asked very much in the exam, that is what is the effect of what is the effect on respiratory system and cardiovascular system on exercise. Okay, so this is a punched question of two chapters, that is how your respiratory system will adapt, and then how your cardiovascular system will adapt. Here I will be taking with the respiratory system. CVS part will be taught by some other person. Okay, so let's begin. First, I will be telling a bit about the basic anatomy. Before that, I just want to tell you one thing. Respiration is a two type. One is the internal respiration, that is number one, internal respiration, and number two, that is the external respiration. What is external respiration is that very much simple. The thing that we're breathing means you are inspiring O2 rich air and you are exhaling out CO2 rich air. That is only external respiration. Okay. And what is internal respiration? Internal respiration is nothing but at the alveolo-capillary membrane. Okay, that I will be talking in the further as we go in this video. I will talking about it further. So there we will see there is the actual exchange of gas, means between the blood and the alveoli. Means oxygen will be delivered to the tissues, and uh, the carbon dioxide that is produced by the metabolism of tissues, that will be uh, taken back by the blood to the alveoli, and here the exchange of gas will take place. Okay, so again I am repeating this. External respiration, that is the you are breathing in and out, that is only external respiration. Internal respiration is that the O2 that you are inspiring. Okay, in external respiration the O2 you are inspiring, that is reaching into the different tissues. Okay, and that O2 is utilized to produce energy, to produce whatever required by the cells. The blood also collects the carbon dioxide that is released as a byproduct of the metabolism of the tissues. Okay, so this is called internal respiration. Now I will be talking a bit about the anatomy. First, let's see. The respiratory tract is divided into two parts. Okay, respiratory tract divided into two parts. One is the upper and another is the lower. Overall, this respiratory tract, it actually extends from the nostrils. Nostrils means the nose. Okay, the nose. From there, it extends to the lung alveoli. Alveoli, I will talk further what is alveoli. Okay, so the upper respiratory tract and lower respiratory tract, it is divided. Upper respiratory tract, it extends from the external layers, okay, means the nostrils, okay, to the vocal folds, okay, and the lower respiratory tract, it extends from the trachea to the alveoli. External layers to vocal folds, and here trachea to alveoli. Here the trachea to alveoli. Here I am telling you the divisions. Means after trachea, the air will enter into the bronchi, then into the bronchioles, and then into the alveolar duct, and then into the alveoli. So this is the pathway of air. Here is a very interesting diagram. You can see here. This is called the Weber's model of tracheobronchial tree. Okay. So it says that the respiratory tract is divided into means the tracheobronchial tree is divided into total 23 generations. Means there are 23 divisions of the tracheobronchial tree, and it is in this also there is another division which is called the conducting zone and the exchange zone. The 1 to 16 generation will be called the conducting zone. Okay, 1 to 16 generation will be called the conducting zone, and the last 7 generation 7 to 23, this will be constituting the exchange zone. Means conducting zone, why it is the name conducting? Because simply it is conducting the air. Like this air will enter from here, it will go here, and like this it will go on. And exchange zone, why it is named exchange zone? Because here actually the exchange of carbon dioxide and O2 takes place between the blood and the respiratory tract, you can say, or the alveoli mainly. Okay. After this, I'll be talking about the alveolo-capillary membrane. Okay, this is a very important short note for your exam also, and very interesting topic. So, alveolo-capillary membrane. You can see, suppose this is an alveoli. This is an alveoli. Okay, this is the alveolar duct. You can see this is the alveoli duct, and this one is the alveoli. Alveoli is nothing but just a sac-like structure at the end of the tracheobronchial tree. Okay, so and these alveoli are very much close with the blood vessel. Suppose this is the blood vessel and this one is the alveoli. So they are very close. And here the exchange of gases will take place. But before knowing how the exchange of gases will take place, let us know the layers. Okay. So you can see this portion. Okay, this portion, there are many layers which are separating this blood from the gas. Here gas is present, O2 rich gas. Okay, and here blood is present, blood. So you can see that these layers are separating the blood from the gas. So what does this layer constitute? First, the layers, I'm telling the layers of the alveolo-capillary membrane. First will be the capillary endothelium. Okay, capillary endothelium means what? Suppose this one is the capillary, I'm drawing it a bit larger, and here the lining of the capillary, there will be endothelial cells. This is called the endothelium. So it is a capillary endothelium. After that, these endothelium are lying on a basement membrane. As you can see, all tissues are lying on a basement membrane. This is the basement membrane. Let me write it down. After that, between this capillary and the alveoli, okay, there is a bit of connective tissue. Okay, so what is the third layer will be connective tissue. Okay, first let me tell the next layer, then I will be telling about the connective tissue. Okay, the next layer will be the basement membrane of the, similarly as I said about the capillary, now the basement membrane of the, what is this, alveoli. Okay, so again another basement membrane. This connective tissue is present between both the basement membrane. Okay, connective tissue is present between both the basement membrane. Another very important thing, this layer 2 and layer 4, okay, means the basement membrane of the capillary endothelium and the basement membrane of the alveoli. These are fused, okay, these are fused at certain places. This fusion, fused at certain places, this fusion actually minimizes, means it minimizes the thickness of the alveolo-capillary membrane. So suppose this is the membrane, since they are fused, this connective tissue layer is not coming into action. Okay, so they are fused, means gases can directly pass through these fused places. Okay, so actually since they are fused, now the distance for diffusion, means the gases that will be diffusing from the alveoli to the blood vessel. Okay, this will be reduced. Now the next layer, you can guess it already, the alveolar epithelial layer. Means after the basement membrane, obviously there will be epithelial layer, and there will be another layer that maximum people forget to write in the exam. That is a fluid. Okay, along the epithelial lining. Okay, means the epithelial lining. Suppose this is the epithelial lining, epithelial lining, epithelial lining. Okay, here there will be some sort of fluid. Okay, this will be a fluid. This is the fluid. Here we will write layer of fluid lining the epithelial layer. If you are given the short note, I think you can write it. You can just draw your own diagram for explaining this alveolo-capillary membrane and just state what are the layers. Here are the layers. Okay. And this fusion part that I told you, you can write this easily. Another thing that I will be telling here only, that is this also you can write in your exams. This alveolar epithelium, okay, alveolar epithelium, it consists of two types of cell. One is type 1 pneumocytes. Another is type 2 pneumocytes. Type 1 pneumocytes are, they are large, very large, flat, and they are only mainly present in the alveolar epithelium. Okay, these are flat and large cells. Okay. Number 2, that is the type 2 pneumocytes. Okay, type 2 pneumocytes, they are thick. They are very much thick. Okay, and they secrete something that is called surfactant. Surfactant, what is surfactant? That we will be talking later. Another thing, what are the other cells present in the alveoli? There can be occasionally macrophages, pulmonary alveolar macrophages, plasma cells can be there, amine precursor uptake and decarboxylation cells. Okay, mast cells, these are also present. APUD that I told, amino precursor uptake and decarboxylation cells. This you may not write on in the exam, so it is not that important. Another thing, suppose these are two alveoli, okay? These are two alveoli. Between the alveoli, there is a connection. There is a connection. What is through a pore? Okay. What is this pore called? Pore. Okay. Pore is called pores of Kohn. This thing is very much important when we will be studying the uh, pathology. Means that is in the tuberculosis. You will see the teachers will be taking this name many times. Okay. So this is very much important for your pathology part that we will be studying later. Now I will be telling you a bit about the pulmonary circulation. Here, what is the pathway of blood? Okay, from heart to lungs and again from lungs to heart. Okay, suppose let us consider this is my heart. Okay, these are the atrium, this is the right atrium, this is the left atrium, and right ventricle, this is the left ventricle. Okay, now blood will enter through the SVC and IVC into the right atrium. From here the blood will enter. Okay, then it will go into the right ventricle. Okay, after that it will go into the lungs. After that, it will go into the lungs through the pulmonary trunk. The pathway of blood will be pulmonary trunk. And from here, the pulmonary trunk will divide into two, right and left pulmonary arteries, supplying the respective lungs. There will be further divisions, repeated division, and will give rise to pulmonary capillaries. And these pulmonary capillaries will be lying between the alveoli. Pulmonary capillaries, as I said, that there will be alveolo-capillary membrane, and these pulmonary capillaries will be responsible. Pulmonary capillaries will be a part of the that alveolo-capillary membrane, and there the actual gaseous exchange will take place. After this, the oxygenated blood will form, and this oxygenated blood from the capillaries will be uh, taken into the pulmonary venule, and these venules will join to form four pulmonary veins, and these veins will drain into the which atrium? Left atrium, and then from left atrium to left ventricle and further into the aorta and then into the rest of the body. Why the oxygen tension in the pulmonary vein is not 100 millimeter Hg. Instead, it is 97 millimeter Hg. Here it says that pulmonary veins, the auto tension is not 100 millimeter Hg since it is receiving the blood directly from the lung, so it should be 100%, but it is not so. It falls a bit below, that is 97 millimeter Hg. Apart from the pulmonary circulation, I told you there is another circulation, that is the lungs will also receive oxygenated blood, oxygenated arterial blood, okay, from the bronchial arteries. This will supply the bronchi and the pleura, and they are drained by bronchial veins. These bronchial veins also have to drain into the heart, so it drains by two ways into the heart. One is this azygos vein, 50% of drains into the azygos vein, and the rest 50% will drain to the anastomotic channel, and they will mix with the oxygenated blood in the pulmonary veins. These pulmonary veins will finally drain the left atrium. Now you can see that the blood that was coming into the pulmonary veins was fully oxygenated. However, it is also receiving some amount of deoxygenated blood from the bronchial veins that comes from the bronchial arteries. This is the reason why the O2 tension does not remain to 100% and it falls below. Now let's get into the next topic, that is pulmonary circulation. Here, one thing I want you to consider is that blood flow in the upright lung. Okay, means suppose this is the lung and this lung is upright. It is influenced by gravity. That is at the level of the right atrium. Means suppose here is the right atrium. This is the right atrium. At the level of the right atrium, the pulmonary arterial pressure. Here the pulmonary at the level of right atrium, the pulmonary arterial pressure is about 20 centimeter of water. Okay, and this is at the middle of the lung. Okay, the pulmonary arterial pressure. Okay, and from this point the arterial pressure will decrease towards the apex, means as we go up it will decrease, and as we go down it will increase towards the base. This is the apex and this is the base. So towards the base it will decrease, towards the base it will increase. Okay, now it is clear. So suppose we are considering that topmost portion of the lungs. Here, the arterial pressure, that is PA, is almost equal to the alveolar pressure. Okay, here the almost the alveolar pressure is equal to the arterial pressure. Now, in certain cases, when either there is some condition, one is the arterial pressure is reduced PA. In case of what? In case of hemorrhage. And there can be another condition, that is increase in pressure of the alveoli, means alveolar pressure is increased. Which case it will be? It will be positive pressure ventilation. Positive pressure ventilation. Okay. In a patient who is admitted in the hospital, suppose we are giving a ventilation, positive pressure ventilation. In that case, the alveolar pressure will be increased. So in these cases, the alveolar pressure, okay, will be greater than the arterial pressure. So here the pulmonary blood flow will stop due to negative pressure gradient, means the pulmonary blood cannot enter into this region, and this will result into a dead space. This will be called as a dead space. What is dead space? I will be talking about this in detail in later part of my video. So now let's focus on this. In zone 1, I have said what will happen, and let's go into the zone 2. Okay, here in zone 2, uh, here in zone 2 what will happen? Pulmonary arterial pressure, that is P capital A. Okay, this will be greater than pulmonary alveolar pressure, which will be again greater than pulmonary venous pressure. That means there is both arterio-alveolar pressure gradient is already there, and there is also arterio-venous pressure gradient. Means there is pressure difference between the artery and alveoli, and there is also pressure difference between the artery and the vein. However, in this region, the blood flow will be due to the blood flow will be due to the what gradient? Arterio alveolar pressure gradient, and not due to arteriovenous pressure gradient. And in the zone 2 region, the flow of blood is intermittent. Now let's come into the zone 3. In zone 3, both the arterial and venous pressure is greater than the alveolar pressure. Okay. So in this case, the means PA will be greater than PV, that will be greater than PA. Okay. So in this case, you can see this is due to the blood flow will be due to arteriovenous pressure gradient. We will be considering the zone 4. This is the lowest part of the lung, means the base. Here, one interesting thing is that the intrapleural pressure will be equal to 0. In the rest upper part of the lungs, we can see that negative intrapleural pressure. Okay, the negative intrapleural pressure in the upper areas, that is responsible for the extra-alveolar blood vessel to be pulled open. And here in the lowermost parts, there is no outward pull. Okay, so pulmonary blood flow in this region will be decreased. Now let us start with the spirometry. Spirometry is nothing but where we measure the lung volumes and capacity. The simple difference between volume and capacity is that capacity is the sum of multiple volumes. Okay, just remember this much. Capacity is just the sum of different volumes. First, let us start with the different volumes. That is, first is the tidal volume. Okay, tidal volume. It is about 500 ml. 500 ml. This is nothing but this is the normal volume of air that is inspired or expired during normal breathing. That is when you are inspiring or expiring normally, the amount of air that we inhale or exhale, that is only the tidal volume. Okay, now is the inspiratory reserve volume, IRV. This is about 2 to 3 liters. Okay, so this is the volume that can be maximally forcefully inspired after a normal inspiration. Okay, means we have already inspired the tidal volume. After that, okay, we have already inspired the tidal volume. After that, if we are trying to inhale much more, okay, that means forcefully you are trying to inhale. After that, whatever volume of air that we can take in, okay, that is after normal inspiration, that will be called as the IRV, inspiratory reserve volume. Now similarly there will be ERV, that is expiratory reserve volume. Okay, this definition is also very clear. First let us know the volume. This is about 1300 ml. Okay, okay. This is also same as IRV. IRV only. Thing is, it is opposite. Okay, that is, it is the volume of air that can be maximally expired after a normal expiration. That means, suppose I have taken in tidal volume and I have expired it. Even after expiring the tidal volume, if we can expire more volume, that will be if when we are expiring more volume, means after the tidal volume we are still expiring, means we are just exhaling the air out of our lungs. Okay, that will be called as the ERV. Means whatever extra volume that we can expire after expiring tidal volume, that will be expiratory reserve volume. Okay, then now we will be talking about the reserve volume. Reserve volume is the volume of air remaining in the lungs even after forceful expiration. Okay, residual volume is almost 1200 ml. Okay, now suppose I have taken in uh, inspirated reserve volume, then tidal volume. Okay, then I've taken also ERV. Now I'm expiring continuously. Okay, now I'm expiring. I've expired tidal volume, then expirated reserve volume. Then also some amount of air will be remaining in a lung. That is called the residual volume. Okay. One thing important about this is it cannot be measured by spirometer. Can't be measured by spirometer. Okay, this is the thing. Okay, now we have done almost all the volumes. Now let's get into the capacity. First is inspiratory capacity. Okay, inspiratory capacity. It is simply the sum of IRV and tidal volume. Its value is almost about 2500 ml to 3500 ml. These volume values you can remember or you may not remember. These are not very important. But at least remember the volume so that you can calculate the capacities. Okay. Now next is expiratory capacity. Okay. This will be ERV, this one, ERV and TV. Okay, TV. Okay, and the last one will, and this value will be 1800 ml. And last one will be FRC, that is functional residual capacity. Okay, it has a definition, that is, it is the amount of air remaining in the lungs after a normal expiration. Okay, means you have already expired out tidal volume. You have expired just the tidal volume. Okay, so after that also how much you can expire? That will be the expiratory reserve volume and the residual volume. Even after normal expiration, this much amount of air, okay, will remain in the lungs. Do you understand it? Means you have just done a normal expiration. After that, whatever amount of air that will remain in the lung, that will be called as the functional residual capacity. Its value is almost about 2500 ml. Now this is a graph. Let us quickly plot this. Okay, this one. This one is the tidal volume. As you can see already, tidal volume. Okay, tidal volume. Now inspiratory reserve volume. Okay, this one. This is the tidal volume. So this one will be the inspiratory reserve volume. This one. Okay, inspiratory reserve volume. And what will be this? This much amount of air you can extra expect. So this will be expiratory reserve volume, and this one, the amount of air that will be remaining in the lung even after a forceful expiration, that is called the residual volume. This will be this much. Okay. So total amount of air that you can hold in lungs can And you can see that how much air you can hold in your lungs. That is reserve volume, ERV, then TV, then you can hold IRV also. So 6 liter of air, okay, you can hold in total. And now let's write down the capacities. First is these two together will be constituting the inspiratory capacity, that is IRV and TV, and these two together will be constituting the expiratory capacity. And from here to here, okay, this will be constituting the vital capacity, and here this will be constituting the functional residual capacity. Okay, so done with this parameter. Very easy topic. We have been reading this in class 11 also. Now let's get into the next topic, that is time vital capacity. Okay, let me write the definition because this is also asked in the MBBS exams also. This is the time related forceful expulsion of air. That is the amount of air forcefully expired in 1, 2 or 3 seconds after a forceful inspiration. These are also called as the forced expiratory volume. The time related forceful expulsion of air. That means you have inspired forcefully. Now you are doing forceful expulsion of the air in first second, second, and third second. This will be called as the forced expiratory volume or the time vital capacity. And each will be having a value: forced expiratory volume in 1 second, forced expiratory volume in 2 seconds, forced expiratory volume in 3 seconds. These have some clinically importance. Okay. First is, let us say, we have two type of lung diseases. One is obstructive type and another is restrictive type. Obstructive type is in which there is obstruction in the respiratory airway. Means whatever path I have shown you in that diagram, that is obstructed somewhere. Example is bronchial asthma or emphysema. Here the example is fibrosis. In restrictive type, the movement of the lung means the lung inflates and then again it relaxes. That movement will be restricted. Okay, so it is called the restricted type of lung disease. In obstructive type of lung disease, what happens if EV1 means forced expiratory volume in 1 second is reduced, and in this one restrict type, what happens if EV1 here also it is reduced. How to distinguish in between them? In both cases they are falling, okay, means that they are decreased. So how to differentiate? For this we also need another thing, that is forced vital capacity, which is what? This is vital capacity performed forcefully. Now if this is also measured along with this FEV1 and FEV2 and FEV3, all these, then what it is seen that in case 1, then that is in obstructive type, both are reduced. Forced vital capacity is reduced and forced expiratory volume is also reduced. But FEV1 is very much reduced with respect to forced vital capacity. So this is the thing. And in restrictive type of lung disease, what it is seen that here also both are reduced. Okay, forced vital capacity is also reduced and FEV1 is also reduced. But here the ratio is normal. Means if I find the ratio between FeV1 and forced vital capacity, this will be normal. Means they are reduced in such a way that the overall fraction remains the same. That is, the ratio remains the same. So in this way, by finding the timed vital capacity and along with the forced vital capacity, you can understand that the lung disease is of obstructive type or is of restrictive type. Now let us move on to the topic that I have told I will be teaching later, that is the dead space. As I have already said that the respiratory tract consists of the two zones, that is conducting zone and in the exchange zone. You can see here is the conducting zone and in the exchange zone. In this conducting zone, as I already said, that here no gaseous exchange takes place in the conducting zone. So this is considered the dead space. And this dead space will be called as the anatomical dead space because this is already present. This cannot be changed. Okay? This cannot be increased or decreased or something. Means wherever there is conducting zone, there is no exchange of gases taking place and that will be considered as the dead space. Normally its value is about, you have to keep in mind about the values. This anatomical dead space value is 150 ml. Another thing is there that is called physiological dead space. What is the physiological dead space? It is defined as the total amount of wasted ventilation in the lung. That is the amount of air that does not take place in gaseous exchange in the whole lung. Suppose in this case, the air have entered into the exchange zone, but some part of the exchange zone, there is no exchange taking place. This part will also be considered as a dead space because this part is not performing any gaseous exchange. Dead space is actually what? That it does not take part in gaseous exchange. The part of the respiratory tract that is not taking part in the gaseous exchange. So it can be anatomical, as I have already said, in the conducting zone that does not take part in the exchange, and there may be some physiological dead space which is normally not present in an individual. Due to some anomaly, the air of some of the exchange zone cannot cannot be exchanged with the oxygenated blood of the alveoli. Okay, so this will also come under the dead space. Let me write down the definition. Total wasted ventilation in the lung, that is air that does not take part in gaseous exchange. One thing you can say, physiological dead space is equal to anatomical dead space plus any other dead space in the conducting zone. That is called as the alveolar dead space. This dead space can be caused due to any emboli. What is an emboli? Emboli is nothing but a distant thrombi. Now, what is thrombi? We'll be reading this in pathology. You don't have to worry about this. Just know that emboli is something, a mass that is blocking the vessel so that the gaseous exchange cannot take place at that particular place. Let us come into another important topic, which is very, very much important. That is the ventilation perfusion ratio. What is ventilation? Ventilation is the exchange of gas. Ventilation over perfusion. Ventilation depends on the amount of O2 that is entering in the conducting zone or the lungs, and perfusion, it depends on the cardiac output. That is how much blood is present in the artery, veins, and capillaries mainly. Ventilation, it is measured as 4 liter per minute, and perfusion, when it is measured to be 5 liter per minute, same, which is equal to the cardiac output. So if you take the ratio, it is coming 4 by 5, almost 0.8. Now from this, we can just convert that some regional imbalances between the ventilation and perfusion can be present. In the apex of the lung, ventilation is less, but perfusion is even lower. Means at apex, what will happen to the ratio? It should increase. Okay, so at the apex, it is found to be 3.3. What happened at the base? At base, ventilation is increased and perfusion is more greater. So what happened to the ratio? The ratio should fall. It is calculated as 0.6. These values you need to remember. How I am saying that at the apex ventilation will be less and base ventilation will be more. Ventilation actually depends on the number of alveoli present. At the apex, you can see there are less alveoli, so ventilation will be less, and at the base ventilation will be increased. Suppose in an area of the lung there is diminished ventilation. That means ventilation is reduced. So what will happen? Ventilation actually depends on the bronchi. So if the ventilation is reduced, if I can reduce the perfusion also, then the ventilation perfusion ratio, that is imbalance, is occurring, that I can prevent. That means if ventilation is reduced, I will also have to reduce the perfusion in equal proportion so that the ratio is not much hampered. So how can I do that? Suppose due to any cause there is reduced ventilation, how will the perfusion become low? I can do what? Vasoconstriction, so that less amount of blood will flow to the lungs and this ventilation perfusion imbalance will be corrected. In another case, some patient is having hypertension, there will decrease perfusion. So how will the body adjust? The body will simply do bronchoconstriction, and due to this bronchoconstriction there will be decreased ventilation. Again, you can see the ventilation perfusion imbalance is corrected. That is the ratio will remain what I have said, 0.8. Means it will reach 0.8, not exactly 0.8. I can give this sign also. Now one important question is asked here also. Again, explain why type of question. Why is tuberculosis infection occur in the apex of the lung? Now, this question is asked in the explain why type. At the apex of the lung, as I already shown you, that there is very much high ventilation perfusion ratio. That means that the apex of the lung is very much ventilated with respect to perfusion. More ventilated, it means there will be an O2 rich environment, means oxygen tension will be increased. This oxygen tension will increase, so there will be maximum amount of oxygen at the apex. So this will favor the growth of tuberculous bacillus, that is Mycobacterium tuberculosis, will grow here more nicely than in the base or any other part of the lung. So this is clear. Let's start with our next topic, that is compliance. What is compliance? Compliance is the stretchability of the lung or chest wall. Mathematically, it represents the, or it is defined as the change in lung volume by change in transpulmonary pressure, represented as del V by del P, with their respective meanings. Okay, from this question, it can be inferred that when the compliance is high, so suppose I am representing this as C. Now this C is high. Okay. So what we can infer from this, that means the transpulmonary pressure that is required to expand the lung volume, that will be less. So in this case, in which the compliance is greater, del P will be less with respect to del V. Del V will be more. Means the transpulmonary pressure that will be required to expand the lung will be very much less. Similarly, when the compliance is low, now suppose the C is low, so that means a greater transpulmonary pressure will be required or must be generated to expand the lung during inspiration. Means del P will increase and del V will decrease. Means a greater amount of transpulmonary pressure will be required to expand the lung during inspiration. There are certain factors, okay, on which compliance depends. Factors on which compliance depends. First is the elastic properties of the lungs, which further depends on the presence of elastin and collagen fibers in the lung tissue, and also depends on their disposition, means conformation, how these elastin and collagen fibers are disposed. Another factor on which it depends, that is surface tension. The surface tension is a detailed topic I will be explaining just after this, but just remember that compliance will depend on the surface tension also. How it depends on surface tension? The surface tension of the fluid that is lining. Remember, I have said that there is also another layer of fluid that lines the alveoli. So that fluid will also exert a surface tension, because of which there will be a centripetal force directed towards the center of the alveoli. That means, suppose this one is the alveoli, this one is the fluid layer over here, and because of this fluid surface tension, okay, there will be a force directed towards the center, and this tends the alveoli to always collapse. Okay, and so this will obviously oppose the expanding tendency or stretchability of the lungs. This surface tension will tend to decrease the compliance. So remember this surface tension of the fluid lining the alveoli. Okay, they will exert a centripetal force towards the center of the alveoli, causing the alveoli to collapse. And because of this collapsing tendency of the alveoli, the stretchability of the lung will decrease, so the compliance will also decrease. There are certain terms related to compliance. Okay, first is specific compliance. This is asked as a short note in your exams, so this is a important topic. Normally, when I am talking about the compliance, okay, suppose if a person has only one lung, we're just assuming, okay, if a person is having only one lung, now his compliance will be half of the person of a normal person who is having two lungs. Similarly, in a elephant, the elephant has huge lung, so in that case, the compliance will be very much high value. And in a small animal like rat, it is much smaller. But in both the cases, the elastic properties of the lungs are very much similar. So to tide over this problem, okay, that means normally the compliance is very much dependent on volume. So to tide over this problem means to exactly show the elastic properties of the lungs, which are not clearly visible in this case, we have proposed a new factor of compliance that is called the specific compliance. What is specific compliance? Specific compliance is normal static compliance divided by FRC. What is FRC? Functional residual capacity. There are many advantages over it. It is a more reliable indicator of the elastic properties. Whereas normally the static compliance only is very much dependent on the volume. And here it is independent of the volume, nearly independent of the volume. It is solely dependent on the elastic properties of the lung. After compliance, as I said you, that I will be teaching the surfactant. So surface tension. I will be explaining the surface tension part, what it is. Then I will be explaining the action of surfactant. So what is surface tension? Suppose this is a container in which water is there. This water has molecules. These water molecules attracting each other with a force known as adhesive force. This is air, this is water. Now at the air-water interface, means this part, okay, interface. Here you can see that above the water molecules there are air molecules, small air molecules, okay, diffused air molecules. Now, what will happen? These water molecules, those are below this, below the interface. They will be attracted by the water molecule below them and the air molecule
above them.
since the attraction between the water molecules is much more greater than with the air, so there will be a net force which will be tending to act downward. That means that the forces between two similar type of molecules is much greater than between air and water molecules. So there is a net force which will be acting downward. So in this case, the surface of the water will tend to contract. That means it will just tend to contract like this. Suppose this is the air water interface and this will contract like this. It tend to contract. Okay. In alveoli, as I have already showed you here in this diagram, you can see this is the fluid, that is green color is the fluid, and the inside part is air, mainly oxygen. You can see I have drawn the lines of forces that will be acting inside. Now surfactant, what is surfactant? These are molecules which reduces the surface tension. It contains phospholipid dipalmitoyl phosphatidylcholine. So big name, but you have to remember this. I can't help you with that. It contains this. Now it is secreted from, remember I have said, secreted from type 2 pneumocytes, and its main function is stabilize the alveoli with accordance with Laplace law. So what is Laplace law? I think this you have studied in physics, and surface tension also you have studied in physics. That is P equal to 2T by R. In respiratory physiology, what is this P? P is the transmural pressure. T is the surface tension. R is the radius of the alveoli. Now suppose one alveoli we have P1 equal to 2T by R1. Another alveoli we have P2 equal to 2T by R2. Suppose I am considering R1 greater than R2. Means this one is the larger one with R1 alveoli and this one is the smaller one with R2. According to this Laplace law, the pressure of the P2 will be greater than P1. Just see the equations, you have done this in physics also. So smaller alveoli, the smaller one, this one, Okay, smaller alveoli will blow into the larger one. But this is not actually what it happens. Okay, the smaller alveoli, those are smaller, that are also stabilized by the surfactant molecules. Now, how the surfactant acts? The surfactant acts by keeping the distending pressure in all the alveoli same. The distending pressure in all the alveoli are maintained by surfactant. Now, how it maintains? Suppose these are the surfactant molecules over here. Okay, I am drawing this. The large one, the surfactant is spread over a large surface area. Means the surface area is also large. Okay, the surfactant molecules are spread over a large area. Their concentration is low. Means area is large, so the concentration of the surfactant molecule is low. Suppose in a water you add less amount of salt, the concentration will be less. More amount of salt, then the concentration will be more. Here, similarly, the concentration of the surfactant in the area will be less because the surface area is large. And in this case, the surface area is small, and the concentration of the surfactant is increased. Here, the concentration of the surfactant is increased. Now, since the alveoli with larger diameter, the surface tension increases as the concentration of the surfactant is low. Whereas in the smaller alveoli, the surfactant molecule concentration is high. There will be low surface tension. Due to this, surface tension radius ratio will be unaltered. The distending pressure in both of them will be same. Okay, means this P2 will be equal to P1 because of surfactant. How we have understood? Means here the concentration of the surfactant will be low. Here the concentration of the surfactant will be high. Since the concentration of surfactant is low here, in this case the surface tension will increase, and here in this case since the surfactant concentration is high, surface tension will be low. In this way, the collapse of the smaller alveoli into the larger alveoli is not occurring because of this surfactant molecules that is produced by the pneumocytes. Now we'll begin with the next topic, that is the transport of gases. First we'll be having the oxygen transport. Here is one law, that is Dalton's law. It says that the total pressure of a gas mixture is equal to the sum of partial pressure of the constituent gases. So from this, the fraction or percentage of the constituent gas is known. Suppose in air there is nitrogen, oxygen and some other gases, and their fraction if we know, then we can simply calculate the total pressure of the gas from the mixture that is in the air. Means we can calculate the partial pressure of oxygen in air, partial pressure of hydrogen in air, partial pressure of nitrogen in air. So this is the utility of Dalton's law. So according to this, it is found that atmospheric pressure at sea level is 760 millimeter Ag, and this contains the gases O2, nitrogen, mainly these two. Okay, and some other gases, CO2 also, water vapor, and other gases. This is all. Okay. Now another interesting thing that I want to consider here, that is dry air. Means the air that we are inhaling. That is, you can see here 160 millimeter of Ag. Means it is the pressure in the dry air. But the alveolar air, the air that is entering our alveoli, that is 100 millimeter Ag. And then further in the arterial blood, that is just after crossing the alveolo-capillary membrane, the pressure falls into 95 to 97 millimeter Ag. Why this is so? Okay, first let us consider from dry air to alveolar air, why it is there is a 60 millimeter pressure difference. First, one important reason is that water vapor. Means as the air enters into the respiratory passage, okay, there occur humidification. So the water vapor that exerts partial pressure of 47 millimeter Ag, and this in turn decreases the pressure of oxygen to 150 millimeter Ag. Thus we can say that humidification in the respiratory passage reduces the partial pressure of oxygen. Secondly, the partial pressure of oxygen in the alveoli is reduced than dry air. So second more important reason is that in the alveoli already existing O2 pore air is present. The alveoli contains air that is very much poor in oxygen, but the air that we are inhaling is rich in oxygen. There is dilution, means the PO2 partial pressure of oxygen is further reduced in the alveoli as very much small volume, that is 350 ml of inspired oxygen mixes with more than 2 liter of alveolar air. Now you can imagine how much dilution it is. This causes dilution. The PO2 falls. I will be explaining the reason why the alveolar air and arterial blood, that is there is difference 3 to 5 millimeter Ag pressure. The reason is attributed to the presence of CO2 and water vapor. CO2 and water vapor causes the partial pressure to fall by 3 to 5 millimeter Ag, and this thing in several steps. Okay, this dilution of air is occurring, means the PO2 is falling in several steps. So all these steps together constitute the O2 cascade. Another thing that I have told you already, this is that this CO2, from where does this CO2 comes that causes this PO2 to fall? Some part of the bronchial veins drain directly into the left atrium, as you know, through the anastomosing channels. And another thing also, in the left ventricle also receives the drainage from the ventricular wall itself. In the left ventricle, where oxygenated blood comes from the atrium, it also gets mixed with the deoxygenated blood which is coming from the left ventricular ventricular wall itself. After supplying the ventricular walls, they come from there to the left ventricle itself. So it is also getting a mixture of deoxygenated with the oxygenated. I will be focusing on this topic, that is transport of O2. Transport of O2 takes place in mainly two ways. Okay, first one is in the physical solution, that is as dissolved in blood, but this is very much insignificant amount. That is 0.3 ml of O2 remains in solution per 100 ml of blood. So you can just imagine how much less it is. But this also has a significance. Why I'm telling this? Because first the O2 that has to enter the tissue actually. First they will not enter directly into the tissue. From the RBC, they first have to come to the solution. That is means in the plasma, physical solution. From the solution, then it has to go to the tissues. That means some amount of O2 must be present in the solution so that it can be transported to the tissue. And obviously, at higher pressure, the amount of oxygen dissolved in solution will also increase. I've talked about the physical solution. Now let's talk about the hemoglobin. It is a protein, mainly a polypeptide, means a heme linked with four polypeptide chains. What is heme and these polypeptide chains, that we will be studying in detail in the biochemistry. So this is a biochemistry topic, the structure of hemoglobin and all. This we will study in biochemistry. What is important to know is each ferrous ion can bind with one molecule of O2. One hemoglobin molecule can bind with how many? Four molecules of O2. This much is clear. That means one molecule of hemoglobin will be carrying four molecules of O2. This is the statement. Now another thing is that is called heme interaction. This is a very much important. This can also be asked in biochemistry, but better to read it in physiology itself. Simply the thing is that when one molecule of oxygen will bind with hemoglobin, then the next molecule of oxygen that will be binding with hemoglobin, its affinity will be increased several times, and this phenomenon is known as heme interaction. So one O2 molecule, after this the affinity will increase several times. This is only the heme interaction. Explanation why it is happening. The four polypeptide chain, that is the globin chain, are bound tightly. That means they are in the tense state, means T state or tense state. When one O2 molecule bind with the deoxygenated hemoglobin, the bonds or salt bridges present in between the globin chains are broken. So if the salt bridges are broken, so they will go from the tense state to relaxed state. As they go into the relaxed state, so the second O2 bind very easily. This is the theory behind the heme-heme interaction that I have discussed. Another important topic, that is the oxygen dissociation curve. Here we will plotting the percentage saturation of hemoglobin on the y-axis, and in the x-axis we will be plotting the partial pressure of O2. So as you can see, this is the sigmoid shape. One thing I want to tell you, that suppose the PO2 is 100 millimeter Hg. Here the hemoglobin will be about 97% saturated. We can see clearly in this curve. Means when the PO2 is 100 millimeter Hg, the hemoglobin will be 97% saturated. Since this curve is like this, means the sigmoid shape is there. If PO2 will fall to 60%, then also you can see that there is not much difference between these 2 points. The percentage saturation only fall to 90%, where it was 97% before. Now it will only fall to 90%. But if we further reduce the PO2, such as say below 60, here the percentage saturation of hemoglobin in the oxygen will be dropping sharply. From these two inferences can be drawn. One is that high altitude up to 10,000 feet and early stages of respiratory disease, in both the cases hypoxia will be absent. Because of this, again I am telling this, at the end of the curve it is almost a straight line, okay, as you can see here. But whenever the PO2 falls below 60, then the percentage saturation will drop sharply. We have to see what happens when we will be shifting the curve to the right and to the left. If the curve will be shifting to the right, that means it will be delivering more amount of oxygen to the tissue, and when it will be shifting to the left, that means the binding of oxygen with hemoglobin will be more. That means less amount of oxygen will be reaching the tissues. This is clear from the diagram itself. Means it will be going like this to the right, and on the left it will be going like this. Let's talk about some factors that will be shifting the O2 hemoglobin dissociation curve to the right. One is increased PCO2. Second is decrease in pH. Third is increase in 2,3-DPG, diphosphoglycerate. Okay. Four is increase in temperature. Let's talk about the 2,3-diphosphoglycerate. It is produced by glycolysis. It is present in abundant in RBC. Because RBC there is excessive glycolysis, so increase in 2,3-DPG. In RBC there is excessive amount of anaerobic glycolysis, the Krebs cycle oxidation, which requires mitochondria. Since mitochondria is absent in RBC, the Krebs cycle also cannot occur. This 2,3-DPG, which is abundant in RBC, will bind to beta chains of hemoglobin. The hemoglobin will be containing mainly the adult hemoglobin I am talking about. It contains two chains, that is one is alpha chain, two beta chain. These 2,3-DPG bind to the beta chain of hemoglobin, and this binding will decrease the affinity of hemoglobin toward the O2. That means O2 will bind less to hemoglobin. So the O2 dissociation curve will be shifting towards the right, as O2 will be binding less to the hemoglobin and the tissue will be having more delivery of oxygen. When the 2,3-DPG level increases, this increases in high altitude, because at that time hemoglobin is inadequately saturated with O2 in high altitude. If the oxygen have to be delivered at the tissues, then the binding must be less. So there is rightward shift, and to explain this we are giving this theory of 2,3 dpg. Similarly, when the tissue pH will decrease, this will also cause the rightward shift of the curve, that is, more oxygen will be delivered to the tissues. This is also known as the Bohr's effect. It's very much important. How this happens? That is, when there is acidosis, H plus ions bind to the histidine residues of hemoglobin. That means H plus ions are produced. This will bind to the histidine residues of hemoglobin, and this causes a conformational change, and the binding of hemoglobin towards O2, Hb to O2 is decreased, so more amount of O2 will be released to the tissues. Another thing that is about the CO2 and temperature, both of them, when they increase, they will tend to increase. Partial pressure of CO2 indicates that there is more amount of CO2 produced at the tissues, so the tissues are undergoing through hypoxia, means there is more of carbon dioxide and less amount of O2. So to overcome this, the body does what? They remove the CO2 and they have to enter the O2 into the tissues that were having a more amount of CO2 previously. When the partial pressure of CO2 in the tissues increases, the hemoglobin curve shifts towards the right. Similarly, it happens along also with the temperature. Now another topic, which is a very small topic that I want to discuss, is myoglobin. Myoglobin is slightly different from the hemoglobin molecule. It takes up O2 from hemoglobin in the blood. It will be taking up O2 from hemoglobin in blood, but it will not release it to the tissue. It will release it to the tissue when PO2 falls to very low level. That is, during, suppose someone is doing exercise or severe exercise, at that time the body's requirement for O2 will be increased and PO2 will fall. So to compensate with that, the myoglobin will release it oxygen. Here you can see the oxygen dissociation curve of myoglobin, which is compared with the hemoglobin. So you can see that it will release at very low partial pressure. This can be indicated from this point, and it is like this. So you can compare it. So let's begin our next topic, that is carbon dioxide transport. Before beginning with the topic, I just want to tell you some things that are very much important. CO2 to tension in arterial blood, this is 40 millimeter Hg. CO2 tension in venous blood, this is 46 millimeter Hg. And arterial content of CO2, that is 48 ml per 100 ml of blood. And the venous content of CO2 is 52 ml per 100 ml. By this, it is implied that the carbon dioxide in arterial blood is less and in venous blood it is high. Obviously, you can understand this because during the tissue perfusion, the oxygenated blood will be entering, mainly the oxygenated blood will be entering the tissue. So from there, they will, after the tissue's metabolism, they will be producing CO2 and that will be returned to the heart by the veins. So obviously, the tension in venous blood will be high and content will also be high. Actual transport of carbon dioxide occurs in three ways. First is physical solution. The next is amino compounds. Another is as bicarbonate. Among these, this one is the most important. Okay, this bicarbonate thing, because maximum amount of carbon dioxide that is transported is in the bicarbonate form. So now, another important information is that 100 ml of blood will transport 4 ml of CO2 from the tissues. How I am telling this? Arterial content is 48 ml per 100 ml. I am telling. Venous content is 52 ml. How much increase of CO2 content is occurring? That is 52 minus 48. So 4 ml of carbon dioxide per 100 ml of blood will be transported from the tissues. Physical solution. About 0.3 ml will be transported per 100 ml of blood in physical solution, the carbon dioxide. How it is present? It is present as dissolved in plasma, and this also acts as a temporary reservoir. The CO2 from the plasma can also enter into the RBCs, or it can also enter into the lungs from this plasma. Just remember this value as carb amino compounds. Here, the carbon dioxide, it will combine with the NH2 radicals of hemoglobin. Hence, the hemoglobin molecule, as I was saying, it is having alpha 2, beta 2. One is for adult. Another hemoglobin is also there, fetal hemoglobin, which is having that alpha 2, gamma 2. And apart from this, also many other types of hemoglobin are there. In fetus, this fetal hemoglobin is present mainly. In adult, alpha 2, beta 2 is predominant. As I said, the carbon dioxide combines with the NH2 radicals of the hemoglobin. So this hemoglobin molecule, alpha 2, beta 2 chain, it will contain the NH2 radicals. With it, the CO2 will combine and it will form what? Carb amino hemoglobin. This name is very much important. Apart from this hemoglobin also, there are certain carb amino proteins and hemoglobin can also bind to them. So these are called carb amino proteins. With this also it can bind, but the maximum portion will be binding with the hemoglobin, since this carb amino proteins in blood is very much less in amount. This carb amino hemoglobin or compounds has a potential to carry 30%. However, it carries only 15 to 20%. What is the reason behind this? The reason is CO2 combines with NH2 radicals. This reaction will be very much slow. However, it has a potential of 30% carrying, but it only carries 15 to 20%. Now the transport of bicarbonate. As you can see in this diagram, which is very much well explained. First, let me tell you. Through this, 70% will be transported. That means almost 3 ml. And the previous one, here it is only 0.7 ml. Now here 70%, that is 3 ml will be carried in this form. First what happens? CO2 can react with water. Then what will be formed? H2CO3. And this will again break to H plus and HCO3 minus. It can happen in plasma and also in RBC. Now, in plasma, this reaction will be very much slow, so it is not of much importance. In RBC, there is an enzyme named carbonic anhydrase. This carbonic anhydrase enzyme will be accelerating these two steps of the reaction. This reaction can occur in a greater speed. Another thing that favors this reaction in RBC is that, so first point I'm telling you that carbonic anhydrase. The next point will be what? Deoxygenated hemoglobin is a proton acceptor. That means it will accept the H plus ions. These H plus ions, this will be accepted by deoxygenated hemoglobin. These two factors will favor the reaction in RBC. Now, why this will happen? The deoxygenated hemoglobin, it is very much stronger proton acceptor. Means it can accept the H plus nicely. As we already know that acidic thing will attract basic things and basic things will attract acidic thing. Now, this deoxygenated hemoglobin is not much acidic. It is also not basic, but it is not also very much acidic. H plus ion being very much acidic, this will be attracted to the deoxygenated hemoglobin. What is chloride shift? Before this, I want to tell you one thing, that when the reaction will be proceeding, that is this reaction, that is CO2 plus H2, H2CO3, H plus CO3 minus, there will be increasing accumulation of HCO3. That is, here you can see in the RBC CO2 is entering. Okay, now see this reaction, CO2, H2O, then H2CO3 will be formed, and after that H plus. All this will be catalyzed by carbonic anhydrase enzyme. You can see this HCO3 minus concentration will be gradually increased in this RBC, and this H plus will be accepted by the hemoglobin Hb molecule. As the concentration increases very much, after some time along the plasma, means between RBC and plasma, there will be concentration gradient of what? HCO3 minus. From RBC, this will come to the plasma, and gradually electrical balance in the RBC will be upset. What will it will be? It will be upset. The HCO3 is moving out at such a great rate. You can see here. To counter this, what will happen? The chlorine will enter. You can see the chlorine is entering here. This chlorine, when it enters the RBC, it will restore the electroneutrality. Electrical balance in the RBC will first get upset due to rapid movement of HCO3 minus from RBC to plasma, and to restore that, Cl minus will enter. This phenomenon is known as what is called chloride shift. This happens in the RBC. Have you seen this ball-like structure? It is known as the band 3 protein. This is very much important. This comes in the MCQs. In PG also they might come. Another thing, similarly, if chloride shift is there, there is something called also reverse chloride shift. Now, what is that? This reaction, whatever it is occurring in the RBCs, this reaction, this full reaction, the reverse thing will occur in the lungs. In lungs, there is HbO2, as the hemoglobin is oxygenated. It is called oxyhemoglobin, and it is very much acidic, and it will release the H plus ions inside the RBC. Now, this H plus ions will be joining with the HCO3 minus, and it will form H2CO3, and from this it will break into CO2 and H2. Just the same reaction in the opposite direction, and here also carbonic anhydrase will work. Now this HCO3 that is formed will enter into the RBCs and again maintain the electroneutrality. Cl minus, what will do? It will move out. So you can see in the RBC the chlorine was entering to maintain the electroneutrality, so it is called reverse chloride shift. And here in case of lungs, the chlorine is moving out, so it is called as the reverse chloride shift. Another topic that is also very much important is Helden effect. Helden effect, simple the statement is release of O2 in the tissues from the HbO2 with formation of deoxyhemoglobin stimulate uptake of CO2 by RBC. That means the O2 that will be carried by the arterial blood will be released in the tissue, and the hemoglobin thus formed will be deoxygenated, and this will in turn stimulate the uptake of carbon dioxide by RBC. Reasons for Helden effect. First is deoxygenated hemoglobin that is formed after the release of O2. This is less acidic, so it is having greater affinity for CO2 as compared to oxygenated hemoglobin. This deoxygenated hemoglobin produced will readily combine with the CO2 to form calm aminohemoglobin. This will help in CO2 uptake. As I said, the stimulate uptake of CO2 by RBC. Another thing is that the deoxygenated hemoglobin, it is again less acidic. It also acts as a strong proton acceptor. As I said earlier also, it will also favor the reaction, this reaction, CO2 plus H2O, H2CO3, H plus, that's HCO3. So this proton will be accepted here. So this will also favor the reaction in the rightward direction. In the lung alveoli, it will be again fully reverse. I'm not going into it. It is simple, very very much easy. You can understand it. If you can understand the Haldane effect, then you can understand the reverse Haldane effect also. So it is also called reverse Haldane effect. This is also very much important. This you can just understand if you understand the Haldane effect. Just like O2 dissociation curve, we also have CO2 dissociation curve. Here we are plotting the carbon dioxide content in the y-axis and partial pressure of CO2 in x-axis. Here also there are certain factors that will be shifting it. As the partial pressure of O2, PO2 will increase, the CO2 dissociation curve will shift right. And the reasons that are explaining the CO2 dissociation curve are Haldane effect and reverse Haldane effect. Another important question, which is very much asked in your explain why type in your MBA exams, that is why there is increased hematocrit in venous blood. As we have already seen the chloride shift and the reverse chloride shift, the number of osmotic particle like Cl minus and HCO3 minus, this will be increased in the venous blood. Why? Because this chloride shift or reverse chloride shift, this will occur after blood have already come out from the tissue that is there in the veins. However, in artery, it is not happening. So the osmotic particles will be increased in the venous blood RBCs. As their osmolarity is increased, so they will what? They will imbibe more water in them, these RBCs, and they will do what? Increase in size. This is one of the reasons. The next reason is that some of the fluid from the artery that are going into the tissues, they are not getting into the vein. They are getting into what? They are getting into lymphatics. Artery to vein, they are not going. They are bypassing the venous system and they are going directly by the lymphatic. And that is why the venous blood will get more concentrated. So this will also contribute to increased hematocrit. This is also asked in the examination, so go through it properly. Now let us begin with our next topic, that is hypoxia. First, I want to clearly state that there are two terms, that is one is hypoxia and another is hypoxemia. These two are very much different. Hypoxia means O2 availability to the tissue is decreased, whereas hypoxia means O2 in the blood is decreased. Hypoxia is of four types, that is first is hypoxic hypoxia, number two is anemic hypoxia, third is stagnant hypoxia, fourth is histotoxic hypoxia. Before going into each of the type of hypoxia separately, let me tell you what are the common symptoms that happens in hypoxia. The main reason of the symptoms that are produced is what you know, it is deficiency of O2. There will be deficiency in energy production, especially in the brain. This will create severe problems. So signs and symptoms are also classified on the basis of three types. First is severe hypoxia. Here what is the importance? It is PO2 will be below 20 millimeter Hg. Where this occurs? This occurs in plane crash at very high altitude. Here what happens? Loss of consciousness in 15 to 20 seconds, and there can be even death in 4 to 5 minutes. Now you can understand that in this case there is very much less chance of life. Second case, that is called acute hypoxia. Here the PO2 will be between 25 millimeter and 40 millimeter Hg. Here what the person will experience having this, first one will be he will behave as if he has consumed alcohol. He will show incoordination, slurred speech, delayed reflexes, euphoria, and subsequently, if this is not treated, in that case, what will happen? Coma, and then death can also occur. So this can be life-threatening if it is not treated. Third type will be chronic hypoxia. So here, first, when it will occur, this is mainly presented case because in the rest of the two cases, the person doesn't survive only. Here, the pressure will be PO2 will be and 60 millimeter Hg between these. So here, what will happen? Intense fatigue and difficulty in breathing. Now, apart from this symptoms, hypoxia also have some signs. Okay, the signs are, number one is cyanosis. Cyanosis I will be explaining in detail in later. Second one is tachycardia. This you must be knowing that tachycardia is increase in heart rate. Okay. And the third thing is hyperapnea. Hyperapnea. Okay. That means increase in respiration, breathing. So now let's discuss the types of hypoxia one by one. First I will take the hypoxic hypoxia. Hypoxic hypoxia. Now what are the causes of it? Hypoxic hypoxia, what are the causes? One is the inspired air O2 tension is low. Example in high altitude. Okay. Next is barbiturate poisoning. Here there will be hypoventilation. And another thing I just want to specify is that, as I said, what is hypoxemia? Okay, this thing. What is hypoxemia? This thing is only present in this type of hypoxia. In this hypoxia, these types of hypoxia, it is not present. Now, the third cause. Remember ventilation perfusion ratio. So third cause will be ventilation perfusion mismatch. It occurs in case of pneumonia, chronic bronchitis, etc. Fourth reason will be defective diffusion. It occurs in pulmonary edema. And another cause can be right to left shunting. Right to left shunting of blood. So these are the five causes. High altitude sickness, I will be talking much more about this in further part of the video. Now, what is the main thing in hypoxic hypoxia? The main thing is that the arterial O2 tension is reduced. That means there is hypoxemia. This is the only characteristic feature of hypoxic hypoxia. So in all these conditions that I mentioned now, in all these conditions, there will be reduced PO2 or arterial oxygen tension. Okay, so this is clear. Now let us go into the compensatory mechanism. Okay, that our body will be adapting, so that this hypoxia can be corrected. Let's start this. Compensatory mechanism to correct hypoxia of this type. First is polycythemia. Polycythemia means RBC will increase. Why it will increase? Due to erythropoietin from what? Kidney. Okay. Second reason is increase in 2,3 DPG. Remember I have said this earlier, and this will shift the O2 dissociation curve, O2 hemoglobin dissociation to the right. And due to this, the affinity of oxygen, the affinity of hemoglobin for oxygen will be reduced. So more O2 will be released at the tissue. Now, the third compensatory mechanism will be hyperventilation. How this happens? The CO2 will be washed out from the alveoli, and the CO2 tension will decrease. And the due to this, as the CO2 tension will decrease, the O2 tension will be increased. So increase in O2 tension due to decrease in CO2 tension. Now, what will be treatment for this type of hypoxia? Treatment is hyperbaric oxygen therapy. Okay. So the treatment is, let's write the treatment. Treatment is O2 therapy. Now the next type, that is you're having anemic hypoxia. Here there is some defect in the hemoglobin or in the RBC. In overall, O2 carrying capacity of blood is reduced. What can be the cause? Decrease in RBC or defect in RBC and defect in hemoglobin or decrease in hemoglobin. So this type of hypoxia is seen in mainly carbon monoxide poisoning. And this is an example of anemic hypoxia. What happens here? Carbon monoxide, this is 230 times more, 230 times uh, more bind has this carbon monoxide has 230 times more binding capacity for hemoglobin than oxygen. Okay, so in presence of carbon monoxide along with oxygen, the carbon monoxide will be uh, having much more affinity toward the hemoglobin, and they will combine totally with the hemoglobin that is present, and they will form carboxy hemoglobin. And very much little amount of hemoglobin will be left out after this for binding with oxygen. So the oxygen will not be able to reach the tissues, and this will result into hypoxia. Here, what will be the compensatory mechanism? Again, compensatory mechanism. What will be those? Again, same. Increase in 2-3 dpg, and it will shift the curve towards the right foot. Another thing is increase in cardiac output, increase in heart rate. Okay, here the main thing is that by increasing the cardiac output, we have to circulate the reduced amount of hemoglobin that is available for binding with oxygen. We have to increase, means we have to cycle the blood more fast. Here the circulation time will be reduced so that the hemoglobin that is required, that will be available for binding with oxygen, can be increased. Okay. So here the objective is to recirculate the quantitatively reduced hemoglobin again and again more frequently. Okay. So that more transport of O2 can occur to the tissues. Another compensatory mechanism will be this again erythropoietin, so that more RBC can be produced to correct this hypoxia. Done. Here, as we have seen that in the previous case, treatment with O2 therapy had worked. See, this I have told. But here in this case, no treatment with O2 therapy. However, it can help. However, if we are giving this patient O2 therapy, it may help, as hyperbaric O2, if you are giving it, it can increase the amount of dissolved O2, particularly in the plasma, and this can be used to replace the carbon monoxide gradually. Okay, so this can be used, particularly the treatment of O2 therapy here can be done with hyperbaric condition. Normal O2 therapy cannot be here enforced because it is not much of that effectiveness. Now the next type of hypoxia, that is stagnant hypoxia. In stagnant hypoxia, let me write it down. Stagnant hypoxia. Here, everything is normal. PO2 and O2 content of blood, everything is normal. Here what happens? The excess of blood, so O2 can't be delivered to tissues. Okay. It is seen in particularly conditions. You have to remember this condition here. One is heart failure, two is venous obstruction. In heart failure, what happens, as you all know, that when the body is not getting proper blood supply, okay, according to its need, that means less amount of oxygen is reaching to the tissue for perfusion. Means with perfusion, less amount of oxygen is reaching to the tissue, so it will give rise to hypoxia. And similarly, in venous obstruction, in particularly venous obstruction, okay, the blood to that particular area cannot come back to heart. Okay, so uh, so there so that portion of tissue will be not be getting proper oxygen, so it will also, there will be also be hypoxia. The next one is the next type of hypoxia is histotoxic type of hypoxia. This is nothing much. Here, the intracellular mitochondria cytochrome enzyme, okay, that is involved in oxygen utilization are ineffective. due to toxic chemicals. So what I said, mitochondrial enzymes are ineffective. There is no O2 utilization. Why it happens? Due to toxic chemicals. Like what? Like cyanide. Okay, these are the two important points that you have to know about histotoxic type of hypoxia. You can very clearly understand by actually you can understand by the name only what it signifies. Okay, suppose here it is hypoxic. Okay, hypoxic, that means it is also having hypoxemia, so it is double hypoxic. Okay, next is anemia. Here it is anemia. Anemia is already present. See, RBC is decreased, HB is decreased, so it is having anemic. Name is having anemic, and is stagnant. There is stagnation of blood, means stasis of blood. Okay, so it is named stagnant. Histotoxic means here the tissue are not functioning. The uh, tissue are not able to utilize oxygen properly due to defective enzymes of mitochondria, and due to certain toxic chemicals like cyanide. Simple. You can just see the name and you can tell what it will be. Okay. So next one topic I was telling about, that is cyanosis. Okay. This is also a very important topic. Cyanosis. 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 What happens? Bluish discoloration of the skin and mucous membrane. Okay. So bluish discoloration of the skin and mucous membrane. Why it happens? Due to reduced hemoglobin in excess. Reduced hemoglobin. I'm not telling the hemoglobin is reduced. I'm telling it reduced hemoglobin is present. Okay. Reduced hemoglobin is present in excess of 5 gram per 100 ml in capillary or arterial blood. Okay. Where the cyanosis thing means this bluish discoloration can be found. The places are lips, dorsum of the tongue, earlobe, nail beds, etc. This topic will be taken in medicine with detailed discussion. Okay. So you don't have to worry much about this. So it is of two types, central, another is peripheral. Here actually this central is mainly due to right to left shunts. Mainly this is only the region, means congenital defects of the heart. Okay. And peripheral, and this is particularly visible in the dorsum of tongue. And rest all other parts also it can be visible. Okay. Dorsum of tongue it is mainly, but rest all part also it can be visible. Okay. And this peripheral is mainly due to vasoconstriction at peripheral parts of the body. Now another important point is it does not occur in anemia. Why? Why it does not occur in anemia? In anemia, in anemia, in anemia there is already, I'm having already reduced hemoglobin. Okay. So if the amount of hemoglobin is low, then how the reduced hemoglobin can be in excess? So as the amount of hemoglobin is already low, so the reduced hemoglobin will also be less. So this does not give rise to cyanosis. Okay. What is the definition? Just check the definition. I've already written. Reduced hemoglobin in excess of 5 gram per 100 ml. If the hemoglobin is only low, then how can reduced hemoglobin become excess than 5 gram per 100 ml? Okay. Do you understand? So in anemia, it does not occur. Okay. Now let's go into the next topic. And that is high altitude sickness. Now, you all have gone to some hill station. Okay. So there this problem arises, particularly in height more than 10,000 feet. Okay, particularly above this height. So you might have experienced some symptoms such as, I'm giving the points: irritability, headache, sleeplessness, nausea and vomiting, Another is dyspnea. Dyspnea is what? Difficulty in breathing. Okay, rest all I think you can understand. Vaishnavi. Okay. So just these symptoms, all the symptoms are mainly due to two factors. Okay, one is cerebral edema, and the another is pulmonary edema. This is very much asked in the question, and this is the most important part. Okay, the high altitude sickness is a very important topic, and it is very much asked in the exam. And these two reasons why the symptoms occurs, it is the most important part of this topic. Okay, so due to hypoxia, okay, there is vasodilatation. Vasodilatation of what? The cerebral blood vessels. Vasodilation of what? Cerebral blood vessels. Okay, so as cerebral blood vessels are dilated, so more blood will be present there. Okay, so this will cause increased capillary pressure also. As the blood vessels are already dilated, this will also result in capillary pressure also, increased capillary pressure. And this increased amount of fluid, okay, from the capillary will be transmitted to the brain interstitium. And so there will be cerebral edema. Now understand this? Now let's go to the second thing. So what will be the reason of pulmonary edema here? Okay, there is due to this high altitude means after going to the high altitude, there is widespread vasoconstriction. Here it was vasodilatation, and here it is vasoconstriction of the pulmonary arteries. So now there will be pulmonary hypertension. However, some pulmonary arteries they will not undergo vasoconstriction at all. They will just transmit the pressure. Instead, they will just transmit the pressure to the capillaries. Now, as the capillaries, the wall of the capillaries are very thin. Okay, so there will be transudation of fluid. Transudation of fluid and even rupture of the capillaries. And they will get into the pulmonary interstitium, so they will cause pulmonary edema. Hope now you can understand this. Now we have understand that symptoms, why these symptoms will occur. We have understood everything. Now the body will also have some compensatory mechanism to correct these changes. This is also asked in the exam also. Compensatory changes of acclimatization. This is a very important topic. This is particularly sometimes asked in the exam as a short note. So you can understand that how much important it is. So first change will be in the respiratory change. First there will be number one will be here first will be hyperventilation. Due to what? Due to stimulation of chemoreceptor. Now, as there will be hyperventilation, there will be hyperventilation very much. So the partial pressure of CO2 will fall as the CO2 will be washed out immediately. Now this will elevate the PO2, means as the PCO2 will fall, the PO2 will increase. This will do what? This will diminish the respiratory drive. Okay. How it will diminish the respiratory drive? As CO2 will be removed from the alveoli, and there this will cause respiratory alkalosis. What is respiratory alkalosis? In respiratory alkalosis, what happens as CO2 is washed out? CO2 washed out. So H plus concentration in CSF will fall. So the hyperventilation that occurred previously, okay, now H plus concentration is falling due to CO2 washout. So this hyperventilation, that is the chemoreceptor that were previously stimulated, will now not be stimulated. So this hyperventilation will stop. However, again, Means again in 4 to 8 days, okay, the hyperventilation will start again and will become about 5 times than normal. And this is caused due to slow pumping out of HCO3 ions from the CSF and brain tissues to the blood. Okay, so the HCO3 ions from the CSF will be pumped out, and CSF and brain will be pumped out from these tissues to the blood, and the pH will be normal, and this alkalosis will not be presented there again, and so the hyperventilation will again start. And moreover, this HCO3 that was formed previously, that is during respiratory alkalosis, this will also be excreted by renal tubules. HCO3 formed will be excreted by the renal tubules. Okay, this much is clear. Now, first we have seen the respiratory change. Now let's get into the next change, that is polycythemia. I have talked about this many times. So there will be stimulation of erythropoietin secretion from the kidney, and this will try to counter this high altitude. Okay, RBC count will increase and they will carry oxygen, and because of this, oxygen supply to the tissue will be increased. Now another is this increase in 2,3-DPG. This also 2,3-DPG. This will increase and this will uh, move the oxygen hemoglobin dissociation curve to the right, and as a result, delivery to the tissues will increase, and affinity of hemoglobin for oxygen will be decreased. Okay. Another thing that is increased is increase in diffusion capacity. Diffusion capacity of O2. Another is the number of capillaries will also increase. That is angiogenesis, and there will be greater diffusion of O2 into the tissues. The mitochondrial enzyme that I talked before, they will also increase. Another thing, apart from this also, those who are living, remember, those who are already living in high altitude, these people, okay, these people will be having very much high total lung capacity. Okay, that is everything. Every capacity will be high, and diffusion capacity of O2. Okay, this will also be exceptionally high. Okay, so this is clear. Now let's get into the next topic, that is decompression sickness. Decompression sickness. Here, here, those people who are doing scuba diving, okay, those people who are doing scuba diving, they have to go deep under the water, and there the pressure is very much high. So they have to ventilate. Point is, they have to ventilate at an increased pressure. And because of this increased pressure of the at which they are ventilating, the alveolar and arterial gas tension are also much higher. So this leads to increase in alveolar and arterial gas tension. So because of this reason, N2 and other gases which are otherwise not dissolved in blood and tissues, they will get dissolved. This N2 is soluble in lipids, okay, soluble in lipid. So mainly in the lipids they will get dissolved. at this high pressure, because of the pressure is very much high. Pressure is high, so they will get dissolved in the lipids. Now these gases are dissolved, okay, in the lipids. Now if the divers who have gone deep into the ocean, if he rises gradually, means slowly he is rising, nothing will happen. But if he rises very quickly, means if rises very fast, then he will be having severe symptoms such as, number one is pain and paresthesia around joints. Then the next will be respiratory distress. Third will be paralysis as because of damage to spinal cord. Okay. So when the driver will ascend very much rapidly, okay, the partial pressure of N2 in the lungs and also in the blood will fall rapidly. As a result, the N2 is liberated in the form of bubbles into the blood and this lipid where they were dissolved earlier. Okay, and these bubbles of N2 that are formed in the tissues around the joints will cause very much pain, and they can even block the small arteries and capillaries in various organs, and they may interfere with the blood supply. So as a result of this, the person will try to increase their respiration. Okay, and so it will be having respiratory distress. They will be having respiratory distress. Okay, and this respiratory distress may go into pulmonary edema, and the person even may die. Okay, and when this paralysis, and now I'm talking about this paralysis, when it will occur, suppose as I said na, as I said that some blood vessels, small blood vessels and arteries will also be blocked, and these suppose these blood vessels are of the spinal cord. So if the blood supply to the spinal cord is blocked, then what it will cause? It will cause paralysis. Okay, now you've understood the reason of these three. In the exam, you have to explain the reason of these 3 points that I have explained just now. Okay, and this this thing that I have told you now, this can also happen particularly in the aeroplanes that aeroplanes that have crashed, okay, at high altitudes. That means normally the aeroplane has pressurized cabins, okay, to protect the passengers from that low atmospheric pressure present outside. Okay, so during the crash, this similar situation can also occur. So this uh, decompression sickness is also known as what? Caisson's disease. Now, what is the treatment? Treatment here is the patient has to be transferred to hyperbaric environment where the pressure is very much high. Okay. Now, from after this, the pressure will be gradually lowered, gradually, not rapidly. Okay. If the rapidity is decreased, then the same symptoms will appear again. The pressure will be dropped gradually from here to there, from high to low. Okay. And the gas bubbles will not form again, and they will diffuse out. Decompression is done at slow rate. Okay. So the patient will show improvement. Regulation of respiration. First, I just want to tell that this regulation of respiration, okay, this is done by two types of control. One is the neural control and another is the chemical control. Before we get into the actual topic, I just want to tell you something that you have seen that when you're sleeping, that time also respiring. When you are awake or doing some heavy exercise, that time you are performing respiration with much more frequency and depth. So this respiration, this can be considered as both as an involuntary and voluntary process. So in Greek mythology, we have an interesting thing that is called as Oindine's curse. Here, there was a person who had done adultery, and as a punishment to him, he was cursed that he will lose the power to breathe automatically. That means he will not be having any automatic control over respiration. He have to continuously breathe and breathe so that he can live. So now you can understand what is the importance of this automatic control. If this is not present, we may die also. Let's get into the topic. The neural control is of two types. One is the cerebral control, that is voluntary, and another is the automatic control. This is involuntary. In the cerebral control, we will be bypassing the pontomedullary centers that are responsible for automatic control and will directly descend to the spinal cord. There they will relay with the lower motor neurons. They will directly supply the muscles of respiration. This voluntary control is very much required during wheeled respiration. That is when we are singing, when we are whistling, when we are even talking also. That time this voluntary control is very much important. Whereas while we are asleep, that time this automatic control is much more important. Let us now talk about the automatic control. The automatic control is done by mainly two centers. One is the medullary center and another is the pontine centers. In the medullary centers, we are having two group of neurons. One is the DRG and another is the VRG. DRG is dorsal respiratory group. The VRG is the ventral respiratory group. There are some points that you have to keep in mind about them. First, it is located near the nucleus of tractus solitarius (NTS). And second point is that it receives sensory input from chemoreceptor, pulmonary stretch receptors, and it will also supply the muscles of inspiration. That is mainly the diaphragm and external intercostal muscles. So this much is enough for the DRG. And now we will be talking about the VRG. So this VRG is further divided into three main areas. The first area is called as the Bodenzinger complex. The next one is called as the pre-Bodenzinger complex. The third one is called as the nucleus retroambigualis. This Bodenzinger complex is primarily concerned during the forceful expiration. This much is enough about the Bodenzinger complex, that it is primarily concerned with the forceful expiration. And the pre-Bodenzinger complex, this is mainly responsible for the pacemaker activity of the respiratory system. That is, this take part in the respiratory rhythm generation. This can be asked in the exams also. This pre-Bodenzinger complex is very much important. The things that I want you to know about this is respiratory pacemaker. Why? Because it takes part in respiratory rhythm generator. This supplies larynx through what? With the glossopharyngeal and vagus nerve. Knowing this much about this is enough. The another thing is this nucleus retroambigualis. This nucleus retroambigualis, it supplies the accessory muscles required during forced expiration. In this diagram, you can clearly see these are the medullary centers. This part is the pons and this one is the medulla. So these are the medullary centers. Here you can see this pink one is the DRG and this one is the VRG. VRG in ventral respiratory group. This one is the Bodenzinger complex. Next one is the pre-Bodenzinger. Last part will be nucleus retroambigualis. So this much is done. Now let's get into the pontine centers. In pontine centers, we're having mainly two centers. First is the pneumotaxic center and the second is the agnostic center. Function of this pneumotaxic center, it is that it will send inhibitory signals or impulses to the pneumatic center, that is this center. So it will make respiration shallow and more rapid. This is one point. And another thing is that it is having a role in switching between inspiration and expiration. That means after inspiration, we have to expand the air. Okay. So in that thing, this pneumotaxic center is having role. It is exactly not determined what role it is having, but it is having some role in switching from inspiration to expiration. So now let's see the function of the agnostic center. When this is stimulated, it will prolong the duration of inspiration and it will decrease the respiratory rate. This much is clear. So this is all about pneumotaxic center and agnostic center. These are all what? They are all pontine centers. Keep this in mind. These centers are very much important. They can be directly asked in the examination also. This is the diagram of the central nervous system. Here you can see this is the cerebrum, this is the cerebellum, and here are the medullary centers and these are the pontine centers. Now in the medullary centers, you can see I have drawn this DRG and VRG, and in the pontine center I have drawn this pneumotaxic and agnostic center. As I have already said, this pneumotaxic center will have some inhibitory effect on the agnostic center. Both this pneumotaxic center and agnostic center will be directly having some influence over this DRG and VRG. And as I have already said you that this DRG situated near the nucleus of tractus solitarius, it will receive the sensory input from chemoreceptors. Suppose these are the chemoreceptors. I am not drawing that. So from this chemoreceptor, this will take some input, and according to those input, if this DRG is stimulated, they can directly send action potential AP to the diaphragm and the external intercostal. So they will contract and they will increase the respiration. So this much is clear how the neural control is done. It is very much simple. You just have to know the names of the centers where which is present and what are the function of each center. This much is very much clear. Okay, this was an interesting topic. Hope you guys have followed this part. Short and long term effect of hypoxia on respiration. As I have already said you that hypoxia will be acting through peripheral chemoreceptor and it will be stimulating the respiration to restore the deficient O2 supply. So it will be acting with the peripheral chemoreceptor and it will stimulate respiration so that the O2 supply will be maintained. But this compensatory mechanism to maintain the O2 supply is short-lived. As due to hypoxia, what it will cause? It will cause hyperventilation. As a result, CO2 will be washed out. The respiration will be reduced. We can say that the short-term measure for correction of hypoxia through chemoreceptor is not very efficient. present. It is less through this peripheral chemoreceptors. But in long term, the compensation for O2 does takes place due to hypoxia.
In chronic hypoxia,
Due to hyperventilation, the CO2 was washing out. This state is reversed slowly with pumping out of HCO3. That is, here HCO3 ions are pumped out from the brain interstitium. So, due to this, pH will return to normal. As the respiratory alkalosis, which is occurring due to increase in HCO3 minus, this is no longer will be present. Due to this, hypoxia will be acting unopposed, and as a result of which there will be stimulation of respiration. Another reason which is attributed to this is the kidney will also excrete this increased HCO3 minus. This will also increase the respiratory drive, as again the pH will return to normal, and this depression of this respiratory alkalosis, that is increase in HCO3 minus, is eliminated. So there will be again unopposed action, and that hypoxia will lead to increase in respiration. Hope it is very much clear. Due to hypoxia, there will be hyperventilation first. I am talking first about the short term. Due to during hypoxia, there will be hyperventilation, so CO2 will be washed out, and there will be depression of respiration. But this is not efficient in long term. That is, in chronic hypoxia, means hypoxia is present for longer time. That time what will happen? HCO3 ions are pumped out. Because of this, the pH of the brain will return to normal, and hypoxia will be continuing. Since the hyperventilation is not taking place, so hypoxia will be unopposed and respiration will be stimulated. Another reason is that kidney will also start to exceed HCO3, and the respiratory drive will be maintained, as the alkalosis of respiration will not be there. The effect of exercise on respiration. We have understood the further topics of this chapter nicely, then this is just very much easy topic. During exercise, what happens? O2 demand will be increased. So first, how this O2 demand is fulfilled? One is by increasing the CO, increasing the cardiac output. Another is O2 extraction by tissues will also increase. During this exercise, large amount of CO2 is also produced in these tissues. That also need to be get rid of. So there is also increase in ventilation. By increasing this ventilation, we are getting more O2 and we are getting rid of this CO2. Another thing that I've told you, in mild exercise, the energy demand will be fully met by aerobic glycolysis. However, if the energy demand in the exercise is excessive, means a burst of energy is needed. For that, the energy is obtained from anaerobic glycolysis. Another is this myoglobin that I have told you for additional O2 into another third point is creatinine phosphate. All these things will supply the extra oxygen during exercise. Since this O2 is utilized, these sources of O2 that were provided, these also need to be replenished again. These O2 that I've taken extra, okay, that I again have to pay off. So for this payment of this extra amount of O2, even after the exercise is stopped, the person will be hyperventilating for some extra time. That is ventilation for some more time, so that the O2 that was utilized from all these three sources, okay, this can be paid off. And this whole thing, that is due to increase in oxygen demand, these three things will happen. And after that also the person will be ventilating just to replenish whatever oxygen he has lost from all these three sources. This thing is overall known as oxygen debt. This comes as a short note in your exam. Chemical control of respiration. So this is done by chemoreceptors. Two types of chemoreceptor. One is central, the another is peripheral. These receptors stimulated by changes in three things. What are those three things? One is PO2, PCO2, and the third one is pH. Let us talk about the central chemoreceptor. Central chemoreceptor are present in the ventral aspect of the medulla, very much close to but distant from the respiratory centers. Now let me show you in the diagram. This diagram that I have drawn, you can see portion is the medulla. It will present almost over here. Okay, see I am marking with a blue color. They will be present. Okay, so what is this? These are central chemoreceptor, very much close to the respiratory center. It is totally different from that. Among all these factors, okay, PO2, PCO2, pH, the main effect is shown by PCO2. That means carbon dioxide tension has the most important effect on respiration. Suppose the CO2 tension in the arterial blood increases, okay, and this will lead to what? Hyperventilation. So what I'm telling is, first there is increasing CO2 tension. Now this will lead to what? Hyperventilation. If there is decreasing CO2 tension, this will lead to what? Hypoventilation. The main regulatory effect of this PCO2 is by the central chemoreceptor. This PCO2 is actually not playing the main role. The main role is again played by this pH only. From where this pH is decreasing, that is how the H plus ion concentration is increasing. How this happen? Again that same thing that I have told you. This CO2 will be combining with water to form H2CO3, and then it will break into HCO3, CO3 minus plus, but it will be H plus. And in this way, this CO2 will be increasing the concentration of this H plus ion. So it will be decreasing the pH. Now understand what I mean. This CO2 gas, okay, is very much diffusible and it can easily pass from blood vessels to brain interstitium and then to CSF, the cerebrospinal fluid. Okay, so since it is very much diffusible, it can cross the blood-brain barrier also, and it can travel from blood vessels to brain interstitium and then into CSF. In the CSF and in the brain interstitium, that is, the CO2, as I said, will be combining with H2O and it will form H2CO3, and this will again dissociate into HCO3 minus and H plus ions. So this H plus ion concentration will gradually increase in brain and interstitial fluid. And since this concentration will increase in very much close vicinity of the central chemoreceptor, and this increase in CO2, as I said, will be very much dependent on this carbon dioxide. In this way, carbon dioxide is very much important. As I was saying, CO2 is very important for regulation of this is PCO2. However, you might argue that this H plus ion that can be produced from different source can also stimulate this central chemoreceptor. However, it is not so. What is the reason behind it? The reason is very much simple. That is, the H plus ion cannot cross the BBB. What is this BBB? Blood brain barrier. So they cannot just directly stimulate the central chemoreceptor. CO2 having the main function on central chemoreceptor and thus it effectively can regulate respiration. CO2 can also stimulate the peripheral chemoreceptor, and the action of CO2 on this peripheral chemoreceptor has much more rapid onset than its effect on the central chemoreceptor. Okay, this thing you should remember that the whatever effect, okay, through this central chemoreceptor CO2 will be having through the peripheral chemoreceptor, okay, the effect will be much more rapid and faster. Now another thing about central chemoreceptor is hypoxia doesn't have much effect on this central chemoreceptor and it has main effect on the peripheral. Through the peripheral receptor it will stimulate respiration. Let's start with the peripheral chemoreceptors. The peripheral chemoreceptors, first one is carotid body and the second one is aortic body. They are located at the bifurcation of common carotid arteries, that is here. You can see this is the carotid artery and this one is the aortic body. As you can see, this is for the carotid bodies. Now this carotid body is composed of two types of cells. One is type 1, another is type 2. These type 1 cells are O2 sensitive. Suppose there is hypoxia, okay. So this type 1 cells, also called as glomus cells, will be stimulated, and from these glomus cells there are certain neurons arising. These neurons will also get excited. How they are excited? It's due to the secretion of chemical transmitter, mainly dopamine. Whatever nerves, those are getting excited. Nerves are actually branches of glossopharyngeal nerve and so will connect to the respiratory centers. They will be directly stimulating these centers, hemotaxic center, state, DRG, all this. Here is the location of the carotid body. See, this one is the carotid body. So here it is located. This carotid body is having the highest amount of blood supply, which is about 2 liter per 100 gram per minute. Okay, highest blood supply it is having. This carotid body's oxygen requirement, it will be met from the dissolved oxygen alone. That means it doesn't have to depend on hemoglobin or something else. Remember, I have taught you that hypoxia, types of hypoxia, among those, specifically in case of anemic hypoxia, this carotid body will be insensitive. But whereas in case of the rest of the hypoxia, like hypoxic hypoxia or histotoxic hypoxia, all those hypoxia, this carotid body will be sensitive. Even in the case of carbon monoxide poisoning, which is itself an example of this anemic type of hypoxia, this carotid body will not be very much stimulated and will not have much role in controlling the respiration. Let me tell you the process by which you will be writing this in the exam. Let's start with the peripheral chemoreceptor. Suppose these are the peripheral chemoreceptor. What I said, mainly the carotid body. Remember one thing, that this aortic body is not having much role. Okay, and another thing here, the type 2 cells present in the carotid body, it is not having much function. Mainly its function is what? Supportive. Means it will just support the type 1 cells. Here, peripheral chemoreceptor, that is the carotid body, as I was telling, they are stimulated by hypoxia. When they are stimulated, what will happen? O2 sensitive K plus channels will be closed. There will be decrease in K plus exit from the cell, and this will lead to what? I think this you have read in nerve muscle physiology and that cardiac CVS physiology also. That if there is decrease in K plus exit from the cell, it will lead to depolarization. Now due to depolarization, what will happen? Ca plus plus will enter, and now there will be, as I said, dopamine will be released by exocytosis. This dopamine released neurotransmitter will be stimulating the afferent nerves of this glossopharyngeal. It will stimulate respiration. Now let's get into the central chemoreceptor. As I already said that this is mainly stimulated by what? CO2. So similar thing, CO2 plus H2O, what it will make H2CO3. I have written this several times in this video. That will form H plus HCO3 minus. This what will happen is that the brain there will be increase in tissue pH, and here there will be increase in CSF pH. This is more important. Now both of these will lead to a respiratory stimulation. Is that clear? This is all about the chemical regulation of respiration.